Today's lecture is the continuation, the second part of the lecture that we had last time, where we spoke about the various reactions, types of enzymes and types of cofactors that go with these reactions in metabolism. I will just remind you that the reason why we're going through these types of reactions and cofactors and enzymes that are connected to them is that these types of reactions, cofactors, enzymes, etc., repeat in metabolism. And once you understand the typical reaction with the typical cofactor, it will be much easier then to see and to remember and to understand the individual steps in metabolic pathways. So that's the goal, and we will continue today with the next set of reactions. And by the end of today, you should have the majority of reactions that we'll see in metabolism. You should have some basic understanding of all of them, all right? So that's the goal for today as it was for before. All right, the next set of reactions, or the next type of reaction that we will talk about is carboxylation. So we will look at reactions called carboxylations. What is a carboxylation? Correct. So it's the adding of a carboxyl group to a molecule. And for carboxylations, we use as the donor of the carboxyl group, we use carbon dioxide, or more specifically, bicarbonate, because carbon dioxide is just a gas, right? But once it dissolves in water, it gets converted to bicarbonate and proton. So usually the actual dissolved donor of this carboxyl group is bicarbonate. So we can have some kind of a compound looking like this, and we add bicarbonate to it. And we get a compound with a carboxyl, okay? This is just a generic way of looking at it, okay? The actual compound may look differently, but the whole idea is that we take bicarbonate, molecule of bicarbonate, and we add carboxy group to a molecule. Conceptually, a relatively simple reaction. Now, what do we need to know about these type of carboxylations? First thing is that this is a reaction that would not occur spontaneously in our cells. So we need something to power it to make sure, remember, it's all about increasing entropy of the universe. So we need some additional process that will sufficiently increase the entropy of the universe so that we can decrease the entropy by performing this reaction. And the stuff that, or the process, the reaction that powers the carboxylation reaction is the hydrolysis of ATP. So we need to add a molecule of ATP, which gets converted into ADP and phosphate. Now, I think often it can be a bit mysterious how this coupling works. How does it work that these two reactions need to occur together? Okay, how is it made sure that this hydrolysis of ATP actually occurs? In this specific case, what happens is that initially bicarbonate will react with one of the phosphate groups of ATP, and we will get a mixed anhydride. Okay, this is just to explain. This is not something that you need to learn, okay, but just to understand how it works. So first, this one phosphate reacts with bicarbonate to form mixed anhydride, and then this mixed anhydride gets connected to the new molecule. There is a cofactor I will mention in a second, but gets connected to this, and the phosphate is released as ADP and phosphate, okay? So overall, we get a hydrolysis and a carboxylation, but they are coupled together because the reaction mechanism requires them to first react together, okay? Just so you understand how this coupling works. All right, so we need ATP to power carboxylations, and as I said, we need a cofactor, and the cofactor which is connected, it's a prosthetic group, so it's a cofactor which is covalently connected to the enzyme. The cofactor that is needed by the majority of carboxylases that we will see in our metabolism is called biotin. It's called biotin. It's a sulfur and nitrogen containing cofactor. I'm not going to draw the structure because we will not look at the specific reaction mechanisms, but if you're interested, it's quite a nice structure of biotin. Biotin is a vitamin Okay, it's one of the vitamins, so we can't synthesize biotin. When later on we talk about vitamins and nutrition, etc. So biotin is required. We need to have it in the diet. But actually, for most people's diets, this is not a problem. So we don't normally see any deficiencies in biotin because there's just biotin everywhere. Okay, but be aware, it is a vitamin.
It's a vitamin too.
Huh?
It's
Vitamin
vitamin 2.
2? What do you mean?
Which number of vitamin?
Oh, okay. Well, they're not numbered, right? They have letters, right? Okay. Okay. So often this would be called vitamin H, okay? But I don't think this is actually useful notation. Just call it biotin, okay? Yeah. Actually, for most of them, if you know the chemical name, that's usually good enough, okay? Now, of course, once you're physicians and you're talking to your patients, you're not going to tell them this is pyridoxine or something. You would tell them this is vitamin B6 or B2. So it's useful for communication, but for biochemistry, it's more important that you know the chemical name of the vitamin rather than, you know, this notation. All right. So biotin is needed for carboxylation. There are carboxylases which do not need biotin, but the majority of the ones that we will see will require biotin. So in your heads, if you see carboxylation, think biotin. It's not 100%, but most of the time you're going to be correct. Good. Now let us, yeah, by the way, which class of enzymes would you think would catalyze this? Ligases. Correct, it would be ligases, because we are forming new bonds between atoms, okay? So they're going to be ligases. Now, let us have a look at the opposite reaction. So we had carboxylation. Now we're going to look at decarboxylation, all right? And this is going to be a bit more complicated because we have several different types of decarboxylations and they all work a little bit differently. You have a question? No, I can.
Is it ligase or ligase?
Ligase, yeah? Okay. So, decarboxylation. Decarboxylation, removal of a carboxy group, nothing mysterious about that. But let's talk about some subtypes of decarboxylation that we will see. One very common type of decarboxylation that we will see is a decarboxylation of an oxo acid. Okay? So we'll see in metabolism a lot of intermediates are oxo acids, and we will decarboxylate many of them. Now, what types of oxo acids we will see in metabolism? Well, we will see alpha oxoacids and beta oxoacids. Okay, so alpha carbon, beta carbon. Okay, we'll see both of these in several different metabolic pathways. Alpha oxoacids and beta oxoacids. Is it clear? Just nod your head if it's clear, the nomenclature. Okay, well, at least some people are nodding, many are not. Yeah, okay, maybe. Good. Yeah, it doesn't work like the more you nod, it means that everyone else is kind of is nodding as well. Anyway, the easier bit about decarboxylation are these beta oxo acids, because beta oxo acids tend to decarboxylate spontaneously. We don't actually have to do anything to them. They will just tend to lose this this carboxy group spontaneously, okay? So that's the easy bit, and again, we'll see it a few times in metabolism. Beta oxoacids tend to decarboxylate spontaneously. They just want to decarboxylate. With alpha oxoacids, however, and that's kind of the trade-off, okay? So with beta oxoacids, it's easy. With alpha oxoacids, it's hard. So alpha oxoacid decarboxylases, so decarboxylation of alpha oxoacids, actually occurs by a different reaction mechanism, which is called oxidative decarboxylation. So when we see an alpha oxoacid, we have to do oxidative decarboxylation, which is basically just a combination of oxidation and decarboxylation. oxidative decarboxylation. Now, this oxidative decarboxylation, once again, a combination of oxidation and decarboxylation, occurs in quite complicated enzyme complexes, which are altogether called decarboxylase, alpha oxoacid decarboxylases, or oxidative decarboxylases of alpha oxoacids. Okay? It's just the name of what they're doing, yeah? Alpha oxoacid decarboxylases. And there are a few of them, and we will see in the very first metabolic pathways, we'll see pyruvate dehydrogenase, we'll see oxoglutarate dehydrogenase. So these are alpha oxoacid dehydrogenases or decarboxylases because they're doing, sorry if this is confusing, why they are called dehydrogenases, because they are oxidizing. They are decarboxylating, and at the same time, they are oxidizing. So they will be called dehydrogenases or decarboxylases. They're doing both of those things.
Could you repeat one more time?
Yeah, okay, so they are called alpha oxoacid decarboxylases or alpha oxoacid dehydrogenases. Their actual names are actually dehydrogenase, but they're doing both of those things. They're oxidizing the molecule and decarboxylating the molecule. You'll see in a second, because I will tell you the actual reaction mechanism step by step. Okay, so they're doing both of those things. They are oxidizing and decarboxylating, so they're called dehydrogenases or decarboxylases. Good. So, as I said, these enzymes are quite complicated. They usually have 3 different subunits. And to make things spicy and interesting, they all contain 5 different cofactors. Okay? So far, we've always seen just 1 cofactor per reaction. Here we have 5 different cofactors. But, as I will show you step by step, it makes sense because each of these cofactors has a specific function, which in the overall mechanism, hopefully, will be clear why they're doing what they're doing. So, now we're talking about oxidative decarboxylation of alpha oxoacids. The first cofactor that we will see in alpha oxoacid decarboxylases is the one which I think is chemically potentially the most interesting cofactor of them all, and it is called thiamine pyrophosphate. Or more correctly, but less often called thiamine diphosphate. So pyrophosphate is the old name for diphosphate. It's the same thing, okay? Usually in textbooks, or most people will use pyrophosphate, but correctly it should be diphosphate, okay? It's the same thing. Now, thiamine diphosphate or pyrophosphate as a cofactor is the derivative of the vitamin thiamine, B1, okay? So it's a derivative of vitamin. And it is also a prosthetic group. We'll see thiamine in several different enzymes, and it's always covalently attached to the enzyme. So it's a prosthetic group, okay? Now, what makes it so interesting? The structure of thiamine, let me just draw it from the paper so that I get all the double bonds correct, looks like this. Nice. The phosphate, there's a positive charge here. There's a hydrogen, which is important, okay? And here is the connection to the enzyme. So this is the structure of thiamine pyrophosphate. Again, this is not something we will expect you to be able to draw, okay? I'm just drawing it so that I can show you what the reaction mechanism is and why this specific cofactor is so interesting and so important. So, as you will see, the ring is quite interesting, right? It's a 5-membered ring containing both nitrogen and sulfur. Does anyone know what the name of the ring is? Thiazole. Okay, so the name of the ring is thiazole. And this is the fact that there is thiazole there is what makes this cofactor actually work. Here is a positively charged nitrogen, and here is sulfur. And in between them, there is a carbon atom having 1 hydrogen atom, okay? Now, what will this positively charged nitrogen and the sulfur do? Well, they will pull electrons towards themselves because they are very electronegative. They want to attract electrons towards them. I mean, obviously not just from here, also from here, but we're interested in specifically in this carbon atom. Because as the electrons are being pulled away, that means that the bond to the hydrogen becomes destabilized, right? Because these atoms are pulling electrons away. So what we get is the hydrogen will get released as a proton. So this thiazole will actually act as an acid. And we are left with negatively charged carbon. Okay? This, for those people who have done more organic chemistry, is quite unusual, right? Carbon doesn't really want to be negatively charged, right? We would see other things, oxygen or whatever, but carbon, it's unusual, okay? But here we have an acidic carbon, basically, because it can release proton and become negatively charged. Any molecule in organic chemistry, any molecule with a negatively charged carbon is called carbanion. Carbanion. And in organic synthesis, it's quite a common thing. It's one way of making complicated compounds, create carbanion, and it will react with all sorts of things. But in our metabolism, it's very unusual because in organic synthesis, you have to use very harsh reagents and very harsh conditions to actually pull away protons from a carbon. But here we have a structure which makes it very easy because the electrons are being pulled away by these two electronegative atoms. And it is this carbanion that allows the first step in our alpha oxoacid decarboxylation, or oxidative decarboxylation. So what happens there is that our alpha oxo, I'll get rid of the proton as well, the alpha oxoacid comes in, just draw it the other way around. And again, I may have mentioned it before or you've seen it before. This carbonyl carbon, the one which is next to the oxo group, is going to be partially positively charged. Why? Well, because the oxygen and the carboxy group are again pulling electrons away from it. So there will be a partial positive charge. Not a real positive charge, but a partial positive charge. Okay, it will be slightly positively charged. Okay? Now, we have a negatively charged carbon here, a partially positively charged carbon here, so they will want to react together. And we will get an intermediate, basically, which looks like this. This carboxy group goes away as carbon dioxide. And instead of this carbonyl atom, we get an OH. So this is what happens in the first step of oxidative decarboxylation of alpha oxoacid. Is it clear what happened there? Okay. Those two carbons are bound together by a double bond. Doesn't really matter very much at this point. Okay. The carboxy group goes away. It's been destabilized by this whole reaction, so it just goes away. And we've achieved the first part of oxidative decarboxylation, which is decarboxylation. Okay. So in the very first step, it's thiamine pyrophosphate, which attacks the carbonyl atom and causes decarboxylation of the acid. That's step one. What happens then? In the second step, we basically do the oxidation. But what is the oxidizing agent in this case? It's the second cofactor that we will see, once again, a prosthetic group, which is called lipoic acid. So this is the second cofactor called lipoic acid. And it is basically just, if you're looking at it and it looks weird, it's actually just a fatty acid with 8 carbons. And 2 of the carbons have SH groups of sulfurs bound to it. Okay? So it's just a modified fatty acid. In fact, lipoic acid is not a vitamin. It's something that our mitochondria are capable of producing. Okay? So this is just a metabolite that we can produce, but it's a cofactor of several different enzymes, including this. It's a prosthetic group. As you can see, it is connected to the protein here through the, normally this would be an acid, but it's connected to an amine and it's connected to a protein. So it's a prosthetic group. So lipoic acid in its oxidized form, and we will see the reduced form in a second, but in its oxidized form comes in and reacts with this intermediate. And what we get is a product which looks like this. Etc., etc. So, the oxidized form of lipoic acid reacts with this intermediate, and what we get is acyl modified lipoic acid, but actually in its reduced form. This is oxidized form, S bound to S, okay? And this is the reduced form, because we have SH, and this would also be SH, okay? But only we have acyl bound to it. So in this step, we've oxidized this weird reaction intermediate to an acyl. Can you see that this is an acyl? Good. So in the first 2 steps, we've already achieved what we needed. We've decarboxylated the alpha oxoacid, and now we've oxidized the product of this decarboxylation to an acyl. Okay. The rest of the steps are basically there just to recover the enzyme. Okay? So these two cofactors, thiamine pyrophosphate lipoic acid, do the main work, and now we just need to recover the enzyme. Because the enzyme has been modified, right? So this has been recovered, but the enzyme is modified on this lipoic acid. So we need to get rid of this and then return the lipoic acid back to its original state. That's what we need to do now. So in the next step, we are going to use another cofactor, but this time it is not a prosthetic group, it's a co-substrate. It's just another substrate of the reaction. And this co-substrate is called coenzyme A. Now, I think I already mentioned it, maybe I didn't. I like to write coenzyme A in this way. So it's coenzyme A, SH, because it's the SH group which will bind acyls. Basically, that's what it's for. Coenzyme A is a transfer, it's a molecule that transfers acyls around, okay, remains of carboxylic acids. And they all bind to this SH group. So in the free coenzyme A, I like to write CoA, SH, because it's free. No acyl is bound to it. So coenzyme A comes in, and basically, the acyl just changed places. It is removed from the lipoic acid, and it is attached to coenzyme A. So what we get from it is the reduced form of lipoic acid. That's easy. And we get coenzyme A with acyl bound to it. Okay, I will actually do it properly. So this is going to be acyl coenzyme A. Now this acyl coenzyme A leaves the enzyme and is one of the products of the reaction. So that's one product of the reaction. But still, the enzyme is modified. It's not still in its original state, right? Because the originally oxidized lipoic acid is now reduced lipoic acid. So in the last step, we need to reoxidize this lipoic acid in order to get the oxidized form, which could then do this reaction. So for that, we will use 2 electron carriers. So it's the last step, but it's actually in 2 steps. The first step uses FAD to FADH2. So it takes 2 electrons from the reduced lipoic acid to reoxidize it back. Now, what do we know about FAD? I will comment on that, whether it's a stronger reducing agent or oxidizing agent. But first, it is always hidden inside the enzyme. So this is not a co-substrate, it's a prosthetic group. Okay? It's part of the enzyme, this FAD. Now, the second part, you'll see in a second that it's a bit more complicated. Okay? So we are still in the enzyme. Lipoic acid has been reoxidized. It's fine. It can work again. But now, we have to reoxidize this FADH2 to return back to the original state. And for that, we're going to use NAD+. Now, I told you in the previous lecture that when we need a stronger oxidizing agent, we usually use FAD or an enzyme containing FAD, because, for example, for turning a single bond to a double bond between 2 carbons, we need something that pulls electrons better. Okay? So this was partially true, okay? Because the actual affinity of FAD to electrons depends on its specific surroundings. Because FAD is always bound to a protein, the type of the protein and the surroundings of the FAD will fine-tune the reduction potential, and we can have different reduction potentials for different FADs in different proteins. So unlike NAD+, which is always free, it's just floating around, okay, that's going to have its reduction potential decided purely by the equilibrium between NAD+ and NADH, okay, that's the only thing. FAD, which is always hidden inside a protein, will get its reduction potential determined by the protein, or co-determined by the protein, okay? So it is not always true that FAD is a stronger oxidizing agent than NAD+. For example, in this case, it isn't, okay? Because we can take the electrons from FADH2 and put them onto NAD+, which, if this were true, what I said before, we couldn't do that, right? We couldn't pull electrons from that. So FAD, that's why I keep emphasizing FAD is always hidden inside the protein, and the protein determines how good an oxidizing agent or reducing agent it is. Yeah? Good. Well, and NADH leaves the enzyme and is the second product of the whole reaction.
So the FAD takes 2 electrons.
Correct.
And the NAD+ takes the electrons from the FAD.
Correct. Yes. Both of them. Yeah,
okay.
Absolutely.
Okay.
So we start with an alpha-oxo acid. We have to put in coenzyme A and NAD+. Right? And we get acyl coenzyme A plus NADH. That's the whole reaction. And carbon dioxide, obviously. That's the whole reaction. But it goes through all of these steps. Okay? Because everything, all of this, is hidden inside the enzyme. Those are not products that leave the enzyme. The only thing that leave the enzyme are the new acyl-CoA, carbon dioxide, and NADH. The rest is hidden inside. Yes?
Which molecule is it that FAD can react with?
It's the reduced lipoic acid. So this is lipoic acid, which needs to be reoxidized to the oxidized form of lipoic acid. They're both lipoic acid. Yeah? This is acyl, yes. Oh, this is S, yes. Sulfur, yeah.
So there's 3 subunits in the enzyme, right? Do all 3 do the same exact thing? Or like does each one have an...
Oh, the 3 subunits in the enzyme, they all do different things. So the first subunit will do this decarboxylation and oxidation and move to lipoic acid. The next subunit takes lipoic acid and moves the acyl to acetyl coenzyme A. And the third subunit reoxidizes lipoate using NADH. So each of these subunits is doing its part in the whole process. Yes?
In the rightmost molecule, it's coenzyme A, and the S is...
Yeah, so coenzyme A, I'm not going to draw the whole structure of coenzyme A because it's quite a big molecule. But the important bit, the bit that actually does anything, ends with an SH group. Okay, so that's where the acyl always binds to the sulfur. Okay, so what we get is actually a thioester. It's like an ester, but it's not an ester caused by a reaction with an alcohol, but it's an ester that is created by reaction with a thioalcohol. Okay, so that's why the sulfur is there, because the acyl is always bound to the sulfur. You don't have to write it there, but I like to write it so that everyone knows that it's bound to the sulfur.
But in the final, acetyl coenzyme A, is the S still there?
I mean, I wrote it like this so that I could fit it into this small space. Okay, but you can absolutely do it. Okay, so you can write something like, if you want.
So it does keep.
Absolutely, yeah. The sulfur is still there. Okay, it's just a matter of whether you write it there or not.
So all the reaction in macro is the carboxylic and then CO. Yeah, it's the all reaction in macro.
Correct.
And the product of the reaction is AC.
Is acyl-CoA. So I wrote it here. It looks like acetyl-CoA. It can be acetyl-CoA if it's pyruvate dehydrogenase, but it can be some other. So it's acyl-CoA, NADH, and carbon dioxide. That's it.
And it's close? No, it's not connected. The acetyl-CoA and NADH.
Those are the 3 products of the reaction: NADH, acyl-CoA, and CO2
And everything else is what happened in the mix.
inside the enzyme. Yep.
How does the NADH get rid of the electrons after?
That's a very good question. What happens to this NADH? It goes into the respiratory chain, into the electron transport chain, and gets reoxidized. And we'll talk about it the day after tomorrow.
So it's all about Krebs cycle.
2 of these enzymes are in the Krebs cycle, or before the Krebs cycle and in the Krebs cycle, but also in other pathways. It's not just about the Krebs cycle. But the NADH will get reoxidized in some other pathway, most likely the respiratory chain, but it could be also used for other things. So the NADH goes away and something else happens to it. And we'll see what happens, or some possibilities of what can happen to it.
NADH and cofactor A are both co-substrates and everything else is processed.
Correct. Coenzyme A, NAD+, are co-substrates, okay? All of the other ones are prosthetic groups, which are inside the enzyme. Yes?
What molecule you said that connected to the CO in lipoic acid?
Oh, so it is the connection of the lipoic acid to the enzyme, okay? It is connected to an amino group of lysine, if you're interested. But it's the connection to the enzyme. Yeah. Yes?
The acid that connects to the sulfur in the lipoic acid doesn't matter how long it is.
Does it matter how?
How long the carbon chain is.
No. It can be any kind of R here. So in the case of acetyl, if this was pyruvate, this would be just CH3, right? Okay, but it could be other things. For ketoglutarate, it's going to be different. For oxoadipate, it's going to be different. Yeah.
So not every carbon chain will become acetyl-CoA.
No, of course not. That's why I'm saying it is acyl-CoA, not acetyl-CoA. Acetyl-CoA is one type of acyl-CoA, but it can be a different type of acyl-CoA depending on which specific carboxylic group you're starting with. Okay? But the reaction mechanism is the same, whether it's pyruvate to acetyl-CoA, whether it's oxoglutarate to succinyl-CoA, whether it's oxoadipate to whatever, I can't remember what the product is. Okay, the reaction mechanism is always the same, but you get different acyl depending on which carboxylic acid you start with. Good. I know this sounds probably a little abstract, but again, already in Krebs cycle, we will see two of these enzymes. And since I don't want to go through the reaction mechanism one by one for each of the enzyme, we're looking at the general scheme, and then we'll just say, okay, this is alpha oxoacid decarboxylase, you know the rest, okay? And it's going to be the same for pyruvate dehydrogenase, for oxoglutarate dehydrogenase, and all the other ones. Good. Shall we take a short break? Short break. Okay, 4 minutes, and we'll continue. Good. All right, there were a couple questions which I think I should probably answer to all of you because they might be confusing. One thing was kind of technical, and that is, the question was, what happens to this part of lipoic acid? Why does it go away? Well, it doesn't go away. I just didn't draw it because it's just connected to the enzyme, okay? It's still there. I just don't care, okay? So I didn't draw it, okay? Because all the interesting stuff is happening here, just so you know, okay? So the change from this to this, I just didn't draw the rest of it, okay? Because it's not important for the reaction mechanism. So that was one thing. The other thing which I can still see that is kind of confusing is that, you know, what is actually happening inside the enzyme and what is happening outside, okay? So, yeah. Yeah, whenever you come in, the camera loses me, so I have to make sure that it's still following me. Okay, good. So the last thing I just want to reiterate is that basically all of this occurs inside the enzyme. The only things that are coming in and out are coenzyme A, which takes the acyl and leaves, and NAD+, which takes 2 electrons and leaves. The rest of it is inside the enzyme. All the other steps happen inside the enzyme. Okay? Good. So this is alpha oxoacid decarboxylase or dehydrogenase. Yeah?
So FAD is present in...
Absolutely. FAD is always in an enzyme. Okay? Never leaves any kind of protein or enzyme. It's always in an enzyme. Yeah? Good. The next reaction is also a decarboxylation reaction, but it works differently, and it will also form a bridge to the next reaction that we will get to. So the next is also decarboxylation, but this time it is a decarboxylation of amino acids. Yep.
So the decarboxylation of alpha oxoacids and beta oxoacids are the exact same?
For beta oxoacids, I mentioned in the beginning, they spontaneously decarboxylate. We usually don't need any kind of enzymes or any other processes. They will just decarboxylate, okay? And we'll see it a few times in metabolism. So the next one is decarboxylation of amino acids. So instead of an oxoacid, we are going to have an amino acid. And for these kind of decarboxylations, we need a new cofactor, a new prosthetic group, which is called pyridoxal phosphate. Pyridoxal phosphate. It is a derivative of pyridoxine or pyridoxal, which is vitamin B6. So it is a derivative of vitamin. We need to get it from the diet. We can't synthesize it. And I will once again draw you the structure of the coenzyme, of the cofactor, which is actually much simpler than the ones that we saw before. But But again, I will just draw it so that I can show you the reaction mechanism. It's not that you need to know the structure of pyridoxal phosphate. So pyridoxal phosphate basically looks like this. So it is a derivative of pyridine, hence the name, pyridoxal phosphate. And the interesting bit is an aldehyde, which is bound to it, hence the name pyridoxal, because it's the aldehyde, right? And then we have some stuff hanging around it. Let's see, there's a methyl group here. And a phosphate here. Anyway, this is not very interesting. The interesting thing is the aldehyde, because that is what allows the catalysis of, in this case, of decarboxylation of an amino acid. So what happens there? In the first step, the amino acid comes into the enzyme. And the aldehyde, the oxo group of the aldehyde, reacts with the amino group. This is a very common reaction. We'll see it many, many times in metabolism. It's a common reaction in organic chemistry as well. When we have an oxo group, an amino group, they will try to react to form an imine. And this is exactly what happens here. So we get an intermediate which looks like this. An imine. Okay, so the oxo group goes away as water, and we get this type of imine structure, a double bond to an amine, to a nitrogen. Yes?
Is the amine the shift base?
We'll get there, okay? So this whole structure, including the ring and including the imine, is in organic chemistry often called a Schiff base, okay? I'm mentioning it because you will find in textbooks, and it's often mentioned, a Schiff base is being formed, but it is just this, having an aromatic ring and an imine formed like this. It's called a Schiff base. For some historic reasons, okay, a Schiff base.
It's hydrogen in the upper
Where? This?
No, on the ring.
This thing? That's a methyl group. This thing? In the middle? Oh, this. Nitrogen. That's nitrogen. It's pyridine. It's nitrogen. It's part of the ring. Yep. Good. So in the first step, we get the formation of this imine, or this Schiff base. And in the next step, because the enzyme is set up in such a way that this formation of the Schiff base and some movements of protons, etc., will destabilize this carboxylic group, and the carboxyl just goes away. Okay? And we've successfully decarboxylated the amino acid. However, you can see that we can't stop there, because the amino acid, or the remains of the amino acid, are still bound to the enzyme. Okay? So we need to recover the enzyme, and for that, we just need another molecule of water, which releases the remains of the amino acid from the enzyme. And what we get is an amine. Okay? A relatively simple reaction mechanism. We will see this type of decarboxylation especially in the synthesis of neurotransmitters, of signaling molecules in the nervous system, where several of the signaling molecules are amines, and we form them by decarboxylating an amino group, an amino acid.
Again, from when the Schiff base is formed, like what happens afterwards, like as soon as
Yeah.
they evolve.
So the Schiff base is formed, and because of the configuration of the active site, this destabilizes the carboxy group on the amino acid, and the carboxyl leaves as carbon dioxide. Okay? And the rest, so this thing, the rest of the molecule, is then released by adding another molecule of water, by hydrolyzing this, and we get the amine. That's basically all there is to it. And that recovers the pyridoxal phosphate. So the pyridoxal phosphate, once again, is an aldehyde. Okay, so we recover the prosthetic group in this case, the cofactor. Yeah? Good. So this concludes decarboxylations. And I will use this mechanism, this reaction mechanism, to get to the next type of reaction, which uses a very similar reaction mechanism. That's why I kind of put it in this order. And the next type of reaction is called transamination. Oh, by the way, when we're talking about decarboxylations, which class of enzymes would you say these would be catalyzed by? Lyases, right? Because what they do is they break a bond between the molecule and the carboxylic group, so generally they would be lyases. However, since classifications are always a bit tricky, for example, the decarboxylases of the alpha oxoacids are actually classified as dehydrogenases, because they are doing both, right? They are decarboxylating and they are oxidizing. So you have to put them in one or the other, right? We can't have them in both classes, so they're actually put into oxidoreductases, okay? But that's just because they're doing two things at the same time. Anyway, let's go to transamination. Transamination is the transfer of an amino group from one molecule to another. Okay, so we take an amino group and move it from one molecule to another. The typical transamination that we see in metabolism, most of metabolism, is taking an amino acid and an oxoacid and swapping the two functional groups, basically. So what we get is, and it's a reversible reaction, so what we get is something like this. Okay? So we take an amino acid and an oxoacid, and we get an amino acid and an oxoacid, but they are kind of flipped around, right? So we're transferring this to this, and here we get an oxo group, basically. Does it make sense, the general idea of transamination? Okay, it's a very important step in the degradation of amino acids. Basically, it happens to all amino acids, so it's a very important reaction. Now, how does it work? What's the mechanism? Well, the good thing is that we've already basically covered the mechanism. The cofactor here, again, is pyridoxal phosphate. And just to give you a kind of a rule of thumb or a hint, whenever there is a reaction which involves amino acids and you don't know what the cofactor is, just guess that it's going to be pyridoxal phosphate. Because pyridoxal phosphate is generally connected to many reactions that use amino acids, okay? So there's a reaction, amino acid goes in and something happens to it, and you don't know what the cofactor is, just guess PLP, because that's what it's going to be, probably, okay? It's very likely. Many reactions with amino acids contain pyridoxal phosphate. So the reaction mechanism is sort of similar, in this case, to our decarboxylation, but in this case, we're not going to decarboxylate the acid, but we're just going to swap the amino group around. So once again, the amino acid goes in and forms a Schiff base, forms this imine structure in the enzyme. That's the same. But in this case, the enzyme is not set up in such a way as to destabilize the carboxyl, so the carboxyl stays. But in the next step, so we need to remove water in order to do this. But in the next step, in the case of transaminases, we get a hydrolysis. So we add another molecule of water, and this Schiff base hydrolyzes, but kind of hydrolyzes in a different way. So it was formed from an amino acid and an aldehyde, but now the bond is cut basically here with the water. So what we get is an oxo acid, and the amino group stays on the pyridoxal. So we get pyridoxamine. This would be called pyridoxamine, right? Do you all see the difference from the previous one? Previously, we said that this carboxyl group goes away. In this case, it doesn't go away, but the next molecule of water, instead of cutting basically, so first we formed this bond, right? But now we're cutting this bond here with the water. The amino group stays with the pyridoxal phosphate, and the oxo group and the oxo acid is being released into the surroundings. Well, we are kind of halfway through because now we have a modified enzyme, so we need to return the enzyme back to the original state. So we need to add our oxo acid. Yeah, it's this one. This oxo acid needs to go in. It forms a Schiff base with this pyridoxamine, basically very, very similar to what we saw before. And then another molecule of water comes in, but this time it cuts this bond, and we are back to where we started. We receive now the new amino acid. Now, this type of reaction mechanism is called a ping-pong mechanism because basically one molecule goes in, gets changed, goes out, another molecule goes in, gets changed, goes out, okay? And it just happens like this. It's like a ping pong, okay? So that's transamination, yeah.
So after the addition of the oxo acid back into the reaction, it's basically for the restoration of the original amino acid?
Yeah, sure. You could call it that, okay? So this new oxo acid acts as an acceptor, sort of, for the amino group, okay? But of course, the whole goal of the reaction is to move this amino group here and this oxo group there. So, but yes, it recovers the enzyme, and we can then use it again for this reaction. Now, as you can see, this is a freely reversible reaction. So we can go from this to that, or from that to this. It's just it can go either way, okay? No special energy or ATP or anything like that is needed for that. Good. Any questions about this? Very common reaction, very important one. If not, there is another reaction that I just want to mention. We'll see it, which is related to amino acids, and that is deamination. This is transamination. So transamination allows us to take an amino group and put it on some other molecule. But there are situations where we need to remove the amino group completely, okay? Not just move it somewhere else, but we need to remove it completely. We will see this in the metabolism of amino acids. As we are degrading proteins and degrading amino acids, at some point, we need to cut this off and turn it into ammonia. And this is what deamination does. So we have an amino acid, and we turn it into an oxo acid and produce ammonia. So this would be called deamination. Now, the most important deamination reaction in our metabolism is deamination of glutamate, of glutamic acid, which occurs by this mechanism. But it is actually oxidative deamination. So not only are we removing ammonia, but we actually have to oxidize the thing first. Okay? I'm not going to go into the reaction mechanism. I don't think it's super important to have that. But just be aware that when we see deamination, specifically glutamate deamination, that's the one that we will see because it's super important for degradation of amino acids. It is, in fact, oxidative deamination, and we actually have to take the electrons away from it so that it deaminates. We can't just cut the amino group out and be done. Okay? We need to oxidize it. Again, if you look at the oxidation state of all those atoms, it makes sense. Okay? This one is actually more oxidized. This molecule is more oxidized than this one. Okay? So we actually have to do that. But yeah, just so you know. So it's oxidative deamination. And as the acceptor for these electrons, we use NAD+, and somewhat bizarrely, we can also use NADP+. Remember, we said that the difference between NAD+ and NADP+ is that they never mix, right? NADP is for anabolism, NAD+ is for catabolism. They will never meet together, okay? Well, yeah. So this enzyme doesn't care whether it's NADP+ or NADP or NAD+. It will just take either. Oxidative deamination. It will take either. Or both. I mean, it can't take both. So yeah, either. Yeah. Of course, yes, yes. Or NADPH. NADH or NADPH. Yeah, it's a promiscuous enzyme that just takes either.
No, it's not. I don't understand. I made it so pretty.
All right. I know it's exciting stuff. Yes.
I just wanted to ask if one of them is used for anabolism and the other is used for catabolism, how can they both be used?
Yeah, that's a good question. How is it possible that if we said that NAD+ is for catabolism and NADP is for anabolism, how can they be used by the same enzyme? Well, they can, okay? That's how it is, okay? So there are a few enzymes in metabolism that will take either of them and they don't care. There aren't very many of them, but this is actually one of them, okay? Good. We're going to move to the next set of reactions. And the next set of reactions are the ones that form esters. So it's going to be esterification. Esters. Now, one of the most common type of esters that we will see in metabolism are esters of phosphoric acid. So, as you know, esters are compounds that are formed by the reaction of an alcohol with an acid. And oftentimes, the acid that we will see is phosphoric acid. Okay, so we will get esters with phosphoric acids, which are called phosphates. And again, many times in metabolism, in glycolysis, not so much in Krebs cycle, but in glycolysis and many other reactions, we are going to have esters with phosphoric acid. And for this kind of esterification, where we are forming esters with phosphoric acid, the usual donor of the phosphate group is ATP. So we're actually taking it from an anhydride, right? You will recall that the three phosphates, or at least the two bonds between the phosphates, are actually anhydride bonds. They are not ester bonds, they're anhydride bonds. So we basically take one of the phosphate groups from the anhydride and bind it to the ester, or bind it to the alcohol to form an ester. So ATP is the usual donor for these kind of esterifications, and this esterification is called phosphorylation. Phosphorylation, and we will see many such cases in metabolism. These phosphorylations are also important for regulating enzymes, for signaling, etc., etc. So it's a very common type of esterification where we use ATP and add the phosphate group somewhere. So it's a phosphorylation reaction. Question? No? The other type of esterification that we will see is a reaction between an alcohol and a carboxylic group, or carboxylic acid. However, in most of these reactions, we don't use actually carboxylic acid to form this ester, but we take a carrier with an acyl. And the carrier is coenzyme A. So we take an acyl coenzyme A, and this will allow us to form an ester. And coenzyme A is released. Okay? So this is a common reaction, for example, in the synthesis of lipids, of triacylglycerols, and many other ones. The one thing in common between these two reactions is that in neither of them, we are just reacting the alcohol directly with the acid. Okay? Here we have an anhydride, that's the donor, and here we have this thioester as the donor. Why am I pointing it out? Both of these formations of esters are thermodynamically unfavorable in our cells. So if we just took phosphoric acid and an alcohol inside our cells, we would not really form any esters of phosphoric acid. So we need some other reaction that will kind of pull it in the right direction. Okay? Here it is basically the hydrolysis. It's not a hydrolysis, but basically the release of ATP, or the change of ATP to ADP that powers it. Here it is the hydrolysis of acyl-CoA, because acyl-CoA also is a macroergic compound. It is kept very far from the equilibrium. So if we hydrolyze acyl-CoA, we also power this reaction by increasing the entropy of the universe. That's why we have these weird donors, because we can't just take an acid and an alcohol and form an ester. I mean, we can do it in a lab, right? If we heat it up and we add some acids, etc., we can actually do that. In our cells, we need some other mechanism to pull these reactions along the right direction. In this case, it is ATP to ADP. In this case, it is the hydrolysis of coenzyme A, because coenzyme A, or esters of coenzyme A, are also macroergic compounds. They also allow the powering of other things. Make sense?
Can we use a thioalcohol?
Sorry? A thioalcohol.
Yeah.
Well, what for? Because coenzyme A is a thioalcohol. Coenzyme A is actually a thioalcohol. So we have a thioester here. Yeah. It's a thioester, is this compound with acyl-CoA. It's thioester. So we kind of trans-esterificate here a bit. Not sure whether we want to complicate it, yeah?
Do we get the same product if we have a thioalcohol and a carboxylic acid, or are we going to form a thioester?
Yeah, I'm not sure where you headed with the answer. So if we had a thioalcohol plus a carboxylic acid, these would not react in our metabolism. Okay? But here they will react with coenzyme A, which is a thioalcohol, but for that we need actually ATP. So if we want to make an acyl-CoA, we need to add ATP for that. We will see that in metabolism, but not now. Okay? What class of enzymes would be doing these kind of things? Transferases, right? We're just taking a functional group from one molecule and moving it to some other molecule. Those are going to be transferases. Good. The opposite direction of esterification is called deesterification. Very good. And deesterification in its, in principle, is hydrolysis. Right? If we go in the opposite direction from a phosphate to a phosphoric acid and an ester, it's going to be a hydrolysis. So we can call it deesterification, that's perfectly fine, but it's actually a hydrolysis. I'm not going to show you any reaction mechanisms. I think they should be obvious, okay? So we just hydrolyze the ester into its constituents. And the enzymes that catalyze it are hydrolases, as you would expect. Hydrolases. Any questions about this? I can see a lot of people are looking a little puzzled. Again, we will see these reactions in those metabolic pathways, and we will talk about them once again, okay? So when we see the formation of acyl-CoA or whatever, we'll say, oh, this is this reaction that we saw before. So it's not the last time you're going to see it, okay? But it's worth getting kind of the basic structure in your heads, because then the things will be hopefully a little bit clearer once you see them in the specific situation. Good. Last few reactions, and we'll go relatively quickly now. Oh, by the way, for these reactions, we don't really need many cofactors, okay? So usually these enzymes do not contain any cofactors. The only thing that I will mention, and that's actually quite important, whenever you have an enzyme that works with ATP, it requires magnesium. So any kind of ATPases, kinases, etc., all the enzymes that work with ATP usually require an ion of magnesium. What is it for? Well, in the structure of ATP, there are a lot of negative charges, right, in the 3-phosphates, a lot of negative charges. And that makes it very difficult for any other molecule to come in and start reacting with those phosphates. So the magnesium just binds to these phosphates and kind of shields the negative charges so that some other molecule can come in and react, okay? So the thing that I want you to remember, whenever ATP is being used for something, usually magnesium is needed for it. Okay? It's not a prosthetic group. It's not really a co-substrate because it doesn't really take part in the reaction in any way. It only acts there to shield those negative charges so that the reaction can proceed. Okay? So this is a connection I want you to keep. ATP, magnesium always go together.
Is it specifically magnesium or any metal?
It's specifically magnesium. At least in our metabolism, it's specifically. I mean, in the lab, you could probably use other divalent cations, but in our metabolism, it's magnesium. Good. Last few reactions. First one is the formation. Yes, don't worry. I mean, that's simple. The next one is the formation of an amide. So to form an amide, we need a carboxylic acid and an amine, or it could be just ammonia. I will just use ammonia to make it easier, okay? Let's use ammonia. It could be an amine, but let's use ammonia. And we get an amide of the acid. We'll see this reaction, I think, only once, if I'm not mistaken. I think we'll see only one such reaction in metabolism, but it's worth knowing because, mainly, it requires the hydrolysis of ATP. So again, it's one of those reactions which we have to couple with the hydrolysis of ATP, otherwise it wouldn't occur. So we need to hydrolyze ATP in order to power this reaction. So it is amidation, or formation of an amide. And again, we'll see one such reaction. When we're forming glutamine, we need this reaction. The opposite reaction is deamidation, correct? Or hydrolysis, we just hydrolyze it. Simple. Yeah. And for that, we need hydrolysis, so different enzyme. Yeah, okay. Last two bits, okay? The next reaction type, which we'll see a few times in metabolism, is called a condensation. A condensation reaction is when two molecules join together and at the same time they release water. We actually saw a few condensations already when we talked about the formation of the Schiff base with pyridoxal phosphate. That was a condensation, right? The amino acid joined with the aldehyde and water was released. It's a condensation reaction. So we can, for example, take this one, but there are many other condensation reactions. So let's have an aldehyde. And we make an amine. Something like this, okay? So it's any type of reaction where we join two molecules together and release water. Often it's going to be, in the reactions that we will cover, often it's going to be an amine with an oxo group, okay, forming an imine. It's a very common reaction that we'll see a few times, okay? It is called condensation reaction. What kind of enzymes would catalyze this kind of condensation? Ligases, yeah? Just ligases, because they're forming new bonds, they're ligases. The final set of reactions, and I will just mention those, not because of the reaction mechanisms, because they are complex, and we'll talk about them in different lectures, but I will just mention them so that we cover all the important cofactors so that we have them all together. So the last set of reactions are reactions which involve the transfer of methyl groups. Transfer of methyl groups. Sometimes these methyl groups are also called C1 fragments or one carbon fragments. Yeah, it's just a methyl group, right? I mean, it doesn't have to be methyl group, as we'll see later on, but they are one carbon fragments and they can be moved around. Now, I will just, as I said, I just want you to know the types of cofactors, mostly prosthetic groups, but one co-substrate, actually two co-substrates, sorry. I want you to know what kind of cofactors deal with these one carbon fragments, okay? Again, we'll not go into the reaction mechanisms, but just so you know the cofactors. So the prosthetic group, which is always a prosthetic group and is needed for some of these C1 fragment transfers, is cobalamin. Cobalamin, which is a derivative of vitamin B12. Now, there are only two reactions in metabolism, in our human metabolism, which use cobalamin. They are super important, and clinically, vitamin B12 and its deficiency, etc., is important. So we will have a special lecture, or actually half of a lecture, talking just about these things. So now just know that cobalamin is a prosthetic group in some enzymes that transfer one carbon groups. The other two cofactors, which also deal with one carbon groups, are co-substrates. So this is a prosthetic group, The other ones are co-substrates. The first one is called tetrahydrofolate or tetrahydrofolic acid. Tetrahydrofolate is a carrier of 1 carbon fragments, and it's a co-substrate. So it receives a 1 carbon fragment and moves it to some other place or to some other reaction. Okay? So it's a co-substrate. Now, I will just say that when we spoke in one of the previous lectures, when we spoke about the first metabolic pathway, not sure if anyone remembers, it was the fixation of carbon dioxide by this really old method, okay? And I told you before that the cofactor on which this reaction happens is called pterin. Now, pterin is a part of the structure of tetrahydrofolate, and all the reactions of tetrahydrofolate with these 1 carbon fragments that we have in our cells are basically exactly the same as in this very old pathway for carbon dioxide fixation. We don't use these pathways for carbon dioxide fixation anymore. We use them as donors of 1 carbon fragments, but the reactions are still the same. So this is actually, this tetrahydrofolate and this 1 carbon metabolism is what we carry from probably the oldest ancestors of life as it existed. The last donor of methyl group is called S-adenosylmethionine. S-adenosylmethionine, a very common donor of methyl groups, of 1 carbon groups. We'll see it in many reactions. S-adenosylmethionine. S-adenosylmethionine. What is it? Well, it's methionine to which we bind adenosine. And the interesting thing is, and that's going to be the last thing I'm going to say, the interesting thing is that we get a positively charged sulfur. It's a sulfonium. This S-adenosylmethionine is a sulfonium compound, which makes it easy for this methyl group to go away and to be donated to something else. Don't worry, we'll see S-adenosylmethionine, we'll see tetrahydrofolate, we'll see cobalamin. Again, this is just kind of the basic structure so that you've heard these things before we get to the specifics. Any questions? Yes? Is what? Yes. Both of these. Just 1 second. Important. This is important. Anyway. These are co-substrates. Co-substrates. Okay, that's it.